Hello everyone, I am Doel Bose Pandey. I am the IVF Lab Director at Indore Infertility Clinic and the Course Director and Trainer at Embryogeny. Today I will be speaking on genetics, the very basics of it. Genetics or genes, chromosomes and DNA are much more beyond just inheritance. Almost all aspects of life are engineered and controlled at a molecular level. So without having a proper understanding of the molecules and the biochemistry between them, we will only have a very sketchy outline of what is happening at a cellular level. So this session will be broken into 4 parts. The first part I will try to cover the basic definitions and basic molecules which take part in genetics. That would be nucleotide, DNA, chromosomes, genes, the central dogma, and the process of translation and transcription. In the second part, we would go through cytogenetics, which would be the study of the structure of chromosomes, chromosomal errors, ploidy errors, SOMI errors, numerical errors, etc. In the third part, we would go through genes and genetic mutation, parts of gene alleles, mutations, etc. And the last part, we would go through epigenetics and divide it into the epigenetic control of a gene expression and types of epigenetic disorder. Let us start with DNA, which is a long linear chain of nucleotides. For the sake of simplicity, we will not go into the basics of the structure of a nucleotide. There are 4 possible variations to a nucleotide in a DNA molecule. They are thymine, cytosine, adenine, and guanine. These 4 possible nucleotides come together in complementary base pairing, where adenine complements with thymine and cytosine complements with guanine, and then they form a classical double helix model of a DNA strand, which is visible on the left of your screen. When you unwind this double helix model, you see a step ladder model, which is visible on the right side of your screen. This is another view of the complementary base pairing, which is visible on the step ladder model. We all know that DNA molecules reside in the nucleus of each eukaryotic cell, and they are visible only during cell replication and division, a structure called chromosomes. Chromosomes are nothing but long, linear and condensed strands of DNA made up of nucleotides, and they are joined by chemical bond by complementary base pairing. Each chromosome contains segments called genes. We will get to the definition of genes in a short while. These strands of DNA molecules are then wound around histone proteins. That gives it a bead on a string appearance, and this compacted spool-like structure is called a nucleosome. These nucleosomes are then condensed to a solenoid structure. The solenoids sit on a protein scaffold to form a familiar structure of a chromosome, which is visible under light microscopy during the prophase of mitosis. What you see on the screen is 3-dimensional view of a DNA double helix present in each cell. Each DNA strand is approximately 1.8 meters long, which is wound on purple histone proteins to form a string on a bead, which then further compacts to a solenoid structure. These solenoids, they come together and compact further to form a familiar structure of a chromosome, which is visible under light microscopy. This is how the solenoid structure finally looks, and this is how chromosomes are segregated and pulled apart into 2 daughter cells during cell division or mitosis. If we have to simplify the concept by use of an analogy, we should consider the entire set of instructions that make us a human or human genome as a book of instruction manual. The 4 nucleotides represented by letters A, C, T and G, when arranged in a meaningful set of individual words, forms a gene that roughly represents a sentence. These sentences, when arranged in a meaningful sequence with correct punctuations and correct paragraph spacing, describe a subset of instructions called a chromosome. So chromosomes are analogous to chapters in the book of genome. So each chromosome represents a separate chapter that houses many genes. Each gene is like a subsection, and a chromosome is nothing but a long linear chain of nucleotides with many genes present on it. Typically, human cells contain about 1.8 meter worth of linear DNA when stretched end to end in each cell, and the chromosomes contain roughly about 19,500 genes on them. So far we have spoken about the structure and arrangement of molecules in DNA and genes and how they are compacted to a compact structure called chromosome within the nucleus. Now we need to understand the purpose of gene. What are the instructions meant for? To understand the central dogma or principle, you have to understand the structure of a protein first. Amino acids are organic compounds that serve as the basic building block of proteins. There are roughly 23 amino acids. When these amino acids combine to form a chain, they are called a polypeptide chain. These polypeptide chains, when they take a specific shape due to presence of various chemical bonds that attract different molecules of the amino acid side chains differently, it forms a three-dimensional structure called a protein. So proteins are like weird-looking three-dimensional blobs of amino acid joined in a specific sequence. The central dogma of molecular biology states that DNA contains instruction for making a polypeptide chain, and the flow of instruction is from a DNA to RNA to finally a polypeptide chain. Now is the time to introduce you to a codon. Each combination of three consecutive nucleotides, that is one of each A, C, T or G, is called a codon or a trinucleotide. What you see on this slide is a double-stranded DNA unwound to a step-ladder model showing a part of the DNA strand. The bottom strand has codons ATG, CCT, GAG and so on. So now we introduce the concept of transcription. Transcription in general means producing a standard text in a language that is understood by all. In our context, transcription is a process of converting the information available in the DNA to a form that is understood by the cell. During the process of transcription, which is beyond the scope of today's lecture, the thymine is replaced by uracil, or T is replaced with U on the mRNA strand. So ATG on the DNA strand is converted to AUG on the mRNA or messenger RNA. mRNAs are single-stranded, by the way. A CCT on the DNA is converted to CCU and so on. So the question is, why do we need all this? What are we trying to achieve? The answer is very simple. We are trying to manufacture a protein by amino acids that are available in the cell's cytoplasm. And for this, we are reading the instructions present on the DNA of our cell's nucleus, transferring the instruction in the form of mRNA to the cytoplasm so that molecular machinery present within the cytoplasm can read these instructions and assemble a polypeptide chain from raw material which is available in the cytoplasm. Translation is essentially the process of converting the triplet codon present on the mRNA molecule to appropriate amino acid side chain. For this, we need to refer to a codon wheel or a codon chart. This chart looks complicated at the beginning, but it is actually not. You have to read the chart from the inner wheel, this is the inner wheel, to the outer wheel. So you read it from A, U, G, A, U, A and so on. Important thing to remember is that AUG corresponds to methionine, which is the start codon. So AUG is methionine, which is a start codon. Similarly, UAA, UAA, UAG, these are both stop codons. So we have three stop codons. The start codon initiates translation, whereas the stop codon stops or terminates the translation process. Another thing to note here is, and remember, is that the triplet codon provides code for roughly 23 amino acids which are represented on the outer wheel. So tyrosines, leucine, proline, these are all amino acids. And there are more than one ways of coding for the same amino acid. For example, proline and leucine have 4 different ways of being coded. For example, CCU, CCC and CCA, CCG, all 4 code for proline. Similarly, 4 ways of coding leucine, 2 ways of coding histidine, and so on. Please remember this information because this will be important when we are talking about genetic mutations. Finally, the polypeptide chain that was just created by the ribosome in the cell's cytoplasm will now be folded to an appropriate usable structure of a protein by the Golgi within the cell's cytoplasm. So far, human genome has been identified with roughly 19,500 genes, give or take a few hundreds, meaning there are at least 19,500 types of proteins that our genome can code for. A quick recap. Present within the nucleus is a double-stranded DNA which is transcribed to create an mRNA. The mRNA is then exported to the cell's cytoplasm via the nuclear pore. The mRNA is then read by the ribosome in cell's cytoplasm, and a polypeptide chain is created using various amino acids present in the cytoplasm. The amino acid chain is then folded to a protein. It is probably worthwhile to show the 3D animation of this entire process to make us appreciate what each cell does in real time. What you see on the screen is the process of gene transcription. So this molecular mechanism is reading the double-stranded DNA and creating a single-stranded mRNA out of it, and the speed is in real time. Isn't this fascinating? Once this mRNA is created, they are exported out of the nucleus through the nucleus gateway or the nuclear pore. This is the exterior of the nucleus, that is basically the cytoplasm. In the exterior, this mRNA strand, I'm sorry for the poor quality of the animation. So what you see is an mRNA strand coming out of the nuclear pore, which is then again read by another molecular mechanism, and a polypeptide chain is being created. So this is a ribosome, and it is assembling a polypeptide chain by reading the triplet codon. Viewing all of this in 3D makes us appreciate what each cell in our body is doing right as we speak. Before ending this lecture, let us go through some translation and transcription facts. A ribosome can join up to 200 amino acids per minute. Proteins occupy up to 40% of cytoplasmic volume in a cell. And mycoplasmas are the smallest self-replicating organisms with the smallest genome. So a total of about only 500 to 1000 genes. And it is the only known cellular organism known to use a different genetic code, that is UGA instead of UGG, as its codon for coding tryptophan. I hope you enjoyed the first lecture, and in the next lecture we are going to cover cytogenetics.